Hello students, welcome to medical lecture series. Today we are going to have another discussion. This is about the classification and the pathogenesis of genetic condition. Generally speaking, we are going to discuss genetic diseases. This lecture series is dedicated to clinical officers and students. My name is Dr. Machel. You are welcome. Don't forget to subscribe and to have a notification on in my channel so that when I update new lectures, you can be updated as well. And also don't forget to share these lectures to your fellow students. In this discussion, my dear students, we'll have the following learning objective. Number one, because we're discussing genetic disease, we'll have to classify them. So it's very important to classify genetic disease because they have classes. And after that, we have to discuss individual class. One of the classes of genetic disease are those genetic disease which arise due to gene mutation. Therefore, we have to describe gene mutation. Why we need to describe them? Because we have seen some of the genetic diseases, they occur due to gene mutation. That's why our second learning objective will be to describe gene mutation. The third learning objective will be classifying disease which fall under category of gene mutation. As we have seen that genetic diseases are classified, and after classifying them, we are starting to discuss one class. After discussing it, we have to mention the disease which follow under those category. After that, we have to describe the pathogenesis of the disease which follow under category of gene mutation. So this is just a highlight of learning objective. So don't worry, we shall discuss them in detail. After that, we have another learning objective. This will be about classification of genetic disease which are due to multifactorial inheritance. What does it tell us? When we are classifying genetic disease, we shall see we have genetic disease which arise due to multifactorial inheritance. Multifactorial means we need many factors, more than one, for the disease to occur. So we have to classify genetic disease with multifactorial inheritance. After that, my dear student, we have to describe the pathogenesis of the disease which fall under category of multifactorial inheritance. As we have seen in the class of gene mutation, we described gene mutation, we classified disease based on gene mutation, and we described the pathogenesis of disease which fall under gene mutation. Now here we are dealing with multifactorial inheritance. This is another category or class of genetic disease. So we have to describe them, to describe the pathogenesis, to mention the disease which follow under category of multifactorial inheritance. After that, we have to move forward and describe genetic disease which arise due chromosomal abnormality. These are disease which occur due to malformation in the chromosome. Malformation in the chromosome, we shall see them. There might be due to abnormal structure of the chromosome, or there might be due to abnormal number of the chromosome. Remember, we shall see in detail every species has has a unique number of chromosome. For example, the human being, we know that it has 46 number of chromosome. So we have to describe genetic disease which arise due to chromosomal abnormalities. And after that, we have to mention to describe the pathogenesis, what happened in the chromosome and leading to manifestation of the disease. So to start, that was a learning objective. Now to start, I would like to say the following as introductory part, that human characteristic, they are transmitted from one generation to another through genetic materials which are known as chromosome. To mention chromosome, I would like to start by drawing. For example, this is a cell. Within a cell, we have a nucleus, and within a nucleus, we have what we call chromosome. We have the chromosome. Within the chromosome, there are very small segments known as gene. they are known as gene. And this gene, they have special code which encode for particular traits, or they carry information about a particular trait or characteristic. And this we say the information is stored in DNA. So within a cell we have a nucleus. Within a nucleus we have a chromosome, and the chromosome they have a smaller segment known as gene, and this gene they have DNA. So generally speaking, traits or characteristic of species, they are transmitted from parent to daughters through genetic material which are known as chromosome. Chromosomes are found within the nucleus, as I have already said. Within the chromosome, we have the small unit, and these units are responsible to code for every protein. When I'm saying every protein, remember we have two types of protein. We have structural protein and we have functional protein. Functional protein, these are enzymes. Of course, remember our body have so many enzymes which are responsible for different functions. So this functional protein are enzymes, while the structural protein are responsible for building up our body. So these information which are responsible for formation of this protein are known as gene. So within the gene we have what we call the DNA. Generally speaking, genes are made up of DNA. So after saying that, I would like to say the somatic human cell has 23 pair of chromosome. When I say 23 pair of chromosome, that means if I multiply by 2, I will have 46. 46, this is the number of chromosome which is found in the human. Among these pair, one pair is responsible for sex determination. That means we have one pair which is responsible for sex determination. If I times 2, that means we have a human has 2 chromosomes which are responsible for sex determination. So out of 46, we remain with 44 chromosome, and these 44 chromosome, they are the one which are responsible for somatic traits. They are responsible for other characteristic which are not sexually determination. They are not responsible to determine the sex of an offspring to be produced. Therefore, generally speaking, the trait of man are governed by a pair of gene. That means one pair from a father and another pair from a mother. So that's why the offspring or the baby which will be born will be having 2 pair of chromosomes. So up to now, I think you have got a concept about genetic and the chromosome which govern traits. So alteration of genes within the chromosome, or change in structure of the chromosome, or change in number of the chromosome can lead to genetic disease. Again, mutation in the gene. Remember we said within a cell we have a nucleus. A nucleus contains chromosome. Chromosome, they have a small unit or segment known as gene. This gene contains DNA, which is responsible to store information for particular traits. So when we have mutation, just the alteration, any change to this gene will lead to discordment of the protein which will be formed, and this will lead to disease manifestation. Also, the chromosome itself, you might not change the gene, you might not alter the gene, but you can alter the structure of the chromosome. To alter the structure of the chromosome, that means you can distort the, for example, this is a chromosome. We know that the human have a chromosome which is double helix. So for example, this is a chromosome looking like that. So you can cut, for example, you can cut a small segment here, or add smaller segment here. So you can add here a small segment. So that means we have this short, with this arm will be very long as compared to this one. This one will be long as compared to this one, and this by so doing you will have distorted the structure of the DNA. Or We know that a human being has 46 number of chromosomes. You can't distort the number. You may add one, or there might be addition or deletion. For example, you can have 47 or 45. These are chromosomes which have abnormal number. So both of them can lead to disease. And this disease which occur due to gene mutation or change in chromosomal structure or change in chromosome number, they are known as genetic disease. They are known as genetic disease. So when we are discussing genetic disease, we are discussing disease which arise due to disturbance of the material which are responsible to carry traits. Remember that the chromosomal pattern in species are very unique, as insisted. A human being has 46. Therefore, because they are very unique, they cannot interbreed and they give an offspring. For example, a human being cannot interbreed with a dog and give an offspring. Why? Because the dog has a unique number of the chromosome and the human being has a unique number of chromosome. So to insist that it's very important that's why you see even a dog can even you force a dog to interbreed with the cow, they will not give an offspring. But those species which are closely related, for example, a buffalo with a cow, buffalo with a cow, buffalo with a cow, when they interbreed, of course they will not give exactly species which resemble as a buffalo or a cow. They will give a species which is not resembling any of the parent. The species produced, they will not resemble either the buffalo or the cow. We say generally all disease inherited as well as acquired. They involve change in gene expression. Hereditary disease. When we say hereditary disease, these are diseases which can be transmitted from one generation to another. And because they can be transmitted from one generation to another, these diseases they occur due to mutation in sex cell. Remember, for a baby or a fetus to be formed, we need the ovum to be fertilized with the sperm. So the ovum is a sex cell. The sperm also is a sex cell. So this cell they have gene. So when you have mutation in the sex cell, we call them gamete. You can lead to a disease, and this disease will be inherited from one generation to another. But when other cells which are not the sex cell, when they are mutated or when there is a structural abnormality in those cells which are not responsible for sex, we have genetic disease. But this genetic disease cannot be transmitted from one generation to another. Why they cannot be transmitted? Because those cells are not responsible for reproduction. For example, you can have a mutation maybe in a thyroid gland. So when you have a mutation in the thyroid gland, the thyroid cells are not responsible for reproduction. So the baby who is going to be born will not have that genetic disease. And this disease remain as acquired, and they just remain to parent, and they cannot pass to daughter. They cannot pass it to the offspring which is produced. In fact, generally speaking, almost all diseases are the result of genetic interaction with other environmental factors. We shall see in the category of multifactorial inheritance that we need more than one gene to be mutated. In addition, we need environmental factors to contribute it. Okay, let us start now our discussion. As I said, genetic disease are grouped. Our first objective was to classify genetic disease. So the first classification of genetic disease are those genetic disease which occur due to gene mutation. They occur due to gene mutation. Remember, gene, this was a small segment of the chromosome responsible for a particular trait. So you mutate that gene. So when you mutate, there is when you mutate, there is going to be a genetic disease. Now the question is, when we are saying gene mutation, what do we mean? When we are talking of genetic mutation, we mean a permanent change of DNA or RNA sequence. Why we say a permanent change? Because normally, do you remember the cell cycle? When the cell is dividing, in the cell cycle we have what we call synthetic phase, that is S phase. In synthetic phase, this is the phase where DNA component are produced, so that when the daughter cells are made, they will have identical DNA as the parent. So during synthetic phase, DNA is being transcripted. That is to make a copy of the DNA. So during the process of making the copy of the DNA, normally so many mistakes do happen. And this mistake, they are corrected. That's why we don't have a mutation. There are some some organisms which they lack this enzyme which is responsible to correct this mistake. For example, HIV virus. The HIV virus don't have an enzyme which is responsible to correct the mistake which occurred due to DNA transcription. Therefore, every species which is produced will have abnormal DNA. That's why the HIV, we are failing to make a drug which can directly kill the HIV virus, because because the virus keep replicating, giving new species which are not identical to the parent. But as far as the human being is concerned, we have an enzyme which is responsible to correct the mistake which occurred due to DNA transcription. So when it happens that these mistakes are not corrected, when it happens that the mistakes are not corrected during DNA transcription, so the daughter cells, they will have those mistakes, and this mistake will become a little copy, so they will become permanent. That's why we say gene mutation means permanent change of DNA or RNA sequence. There must be a permanent. But when they are corrected, so the mistake will not be there, and therefore we cannot say that is a mutation. Mutation is when there is a permanent change in the DNA sequence. Mutation can affect the germ cell again. As I said, when the mutation affects the germ cells are ovum and sperm, and these are responsible for reproduction. Therefore, the baby who is going to be born will have that disease, and this disease will be transmitted from one generation to another, because they have occurred in sex cell, and therefore this disease are known as inherited disease. While mutation in somatic cell, these are cells which are not responsible for reproduction. When there is a mutation in somatic cell, because the somatic cells are not responsible for reproduction, therefore this disease cannot be transmitted to the two offspring. Remember, for example, you have a mutation in a cell which is responsible maybe for a skin formation. So that cell, because it's not responsible because the mutation has not occurred in the sex cell, this mutation cannot be transmitted from one generation to another. This mutation will remain at the causative of cancer. Of course, they remain at the risk for cancer and some congenital malformation, but they cannot be transmitted from one offspring to another. For example, you may have a father who has the extra digit. We normally have 5 digit in our hand. So you can have a parent with the extra digit, maybe 6. But this parent cannot give an offspring, cannot have a baby with 6 digit. Why? Because the cell which is affected is not responsible for sex. That's why you may find the father has extra, but the baby has no extra digit. But if the mutation occurred in the sex cell, that means every offspring will have the same problem, because they will be transmitted from one generation to another. Okay, coming now to classify these genetic diseases. As I said, we have those which occur due to gene mutation. The second, we have those which occur due to multifactorial inheritance. And the last, we have those which occur due to chromosomal aberration. When I say chromosomal aberration, I'm talking of abnormality of the chromosome. Abnormality of the chromosome, as I said, they can be due to structural defect or number. A number meaning that we have more than the number required or less than the number required. So when we are discussing genetic disease, I would like first to start with our first class, that is gene mutation. So welcome to genetic disease which are due to gene mutation. In this point of gene mutation, that means there is a change, permanent change of DNA sequence, and this category they are also divided into two types. We have point mutation. In point mutation, we say these are genetic diseases which occur due to gene mutation, but in a gene mutation there is only a change in a particular point. There is a change of a single nucleotide base by a different base. This will result to replacement of one amino acid by another amino acid. By so doing, you give a protein which is abnormal. Remember, I said within a cell we have what? We have a nucleus. Within a nucleus we have what? We have a chromosome. Within the chromosome we have gene, and this gene they have DNA. Of course, the DNA is a coding. This is information storage, storing code. So when you have a mutation in the gene of point mutation, that is, there is a substitution of one nucleotide base to another. So to understand that, let us have a look of the DNA. The DNA and the RNA, they are both made of nucleotide bases. Nucleotide bases. Do you remember the nucleotide bases? The nucleotide bases, these are, the DNA, they have nucleic acid, and the nucleic acid, they have nucleotide bases. Okay. Do you remember the nucleotide base? We have the adenine, we have the cytosine, we have the uracil, we have the thymine, we have the guanine. So these are nucleotide bases: adenine, cytosine, uracil, thymine, guanine. So when you are talking of point mutation, that is, assume we have this sequence. We have this sequence that in our particular segment, our particular gene, we have adenine. After adenine, we have thymine. After thymine, we have cytosine. After cytosine, we have guanine. I'm not talking of uracil. Remember the difference between RNA and the DNA. The RNA, they don't have thymine, they have uracil. While the DNA, they have thymine instead of uracil. That's what makes the difference between DNA and RNA. They both contain nucleotide bases, but the nucleotide bases, the only difference is that in DNA, thymine is replaced by uracil. In RNA, uracil is replaced, so the thymine is replaced by uracil. That is, DNA have thymine, while the RNA have uracil. So this is our code. Remember, adenine pair with, do you remember? Pair with thymine, while the thymine also pair with adenine. Cytosine pair with guanine. Guanine pair with cytosine. So when we are taking this is information. So when we are interpreting the information, three nucleotide base are responsible for a particular amino acid, single amino acid. Adenine, thymine, cytosine. The arrangement of adenine, thymine, cytosine is responsible for a particular amino acid. So when you change this sequence, for example, when you change the sequence, instead of thymine, I can just say again cytosine. So when cytosine will have to pair with guanine. So the arrangement of adenine, cytosine, cytosine. will not signify the same amino acid as when it was adenine, thymine, cytosine. So you have a different amino acid. And when we are making a protein, amino acid should be arranged in a sequence. There should not be a change in number, in position. For example, we can have amino acid. Let me mention some few amino acid. For example, let's say I have amino acid which is alanine. I have amino acid which is valine. I have another amino acid maybe which is phenylalanine. So this amino acid, another to mention maybe guanine. Okay, those three are enough. They are enough to explain what I want to explain. For assume now, we have the sequence of adenine, guanine, thymine. Adenine, guanine, thymine, maybe they denote alanine, while adenine, thymine, adenine, thymine, they are responsible for valine, and adenine again, guanine, adenine, guanine, they are responsible for phenylalanine. So if the normal sequence was adenine, guanine, thymine, the protein which was to be made should have alanine as the first amino acid to be placed in position number 1. So assume now you do a mutation in this sequence. Now let me change the thymine and replace it with guanine again. Now guanine. The guanine is responsible for phenylalanine. Phenylalanine is another amino acid which is quite different from alanine. So the protein which is going to be formed will have phenylalanine in the first position of the protein. That position will not be function. That protein will not be function. Okay, another example. Let's say I replace guanine. I put thymine. So adenine, thymine. Adenine, thymine was responsible for valine. That means when you are making a protein, the first amino acid to be placed in position number 1 will be valine. It will not be alanine as the correct amino acid. Because you are placing a different amino acid, the protein which is going to be formed will be a dysfunction, will be not working or abnormal protein. Therefore, this is what we call point mutation. In a point mutation, what you just do, you change one nucleotide base. Once we change one nucleotide base, you change the reading code. When you change the reading code, the amino acid which is going to be placed will be different. So when you place a wrong amino acid, you are going to form a protein, whether it is a functional, that is the enzyme, or structural, that is responsible for structure, they will be abnormal protein, and they will lead to genetic disease. Again, if this point mutation occurred in a sex cell, the abnormality will be transmitted from one generation to another. But if this mutation occurred in a somatic cell, the mutation which occurs in the somatic cell, they will not be transmitted from one generation to another. They will just lead to congenital abnormalities and putting a patient to high risk of cancer. So after explaining that, I say these results, the genetic point mutation, they result from a substitution of a single nucleotide base by a different nucleotide base, resulting in replacement of one amino acid by another amino acid, therefore producing abnormal protein. So the mutation giving rise to sickle cell anemia is a classical example When we are talking of anemia, anemia is a classical example of gene mutation which are due to point mutation. Genetic disease, sickle cell anemia is a genetic disease. Is a genetic disease which follow under category of gene mutation. In gene mutation, they follow under category of point mutation. That means we have substituted one amino acid by another. For example, let us have a look of this picture. This is a point mutation which explain how sickle cell anemia or sickle cell disease happen. Normally, this is the DNA. The DNA will be, this is a normal DNA, will be transcribed into messenger RNA. In messenger RNA, look, we have this sequence, guanine, adenine, guanine. Guanine, adenine, guanine is responsible to code amino acid which is a glutamic acid. And this is a glutamic acid in position number 6. Now, when there is a point mutation, remember what we said in point mutation, there is a substitution of one nucleotide base by another. Now, in abnormal beta chain of the hemoglobin, what will happen? That is, it was supposed to be guanine, adenine, guanine. Now adenine has been replaced by uracil, so it is guanine, uracil instead of adenine. So again, guanine. So we have a code which is guanine, uracil, guanine. Guanine, uracil, guanine will code for valine, not glutamic acid. That means in position number 6, instead of placing glutamic acid, we are going to place valine. So when you are going to place valine, that means we are going to form a protein which is dysfunction. That is what happened in position in sickle cell anemia. And this is a classical example of point mutation. Remember, point mutation, we said there is a substitution of a single amino acid by another. The normal was supposed to be guanine, adenine, guanine. So you have substituted adenine by uracil. So by doing so, you are going to code for valine amino acid, which is amino acid, instead of glutamic acid. So in position number 6, you replace valine instead of glutamic acid, and by so doing, you form a protein which is dysfunctional. That's why patient with sickle cell anemia, they have disease because of genetic mutation which is due to point mutation. That is a substitution of glutamic acid by valine in position number 6. Now let us have another look of gene mutation. The gene mutation, we have what we call a frame shift. Frame shift occur due to insertion or deletion. So to understand this, I would like to have a white page which I can. Okay, let us use this whiteboard to explain the frame shift. When we are saying a frame shift, this is a change of reading sequence. Normally, remember, we read nucleotide base in triple. 3 nucleotide base, they denote a single, they code for a particular amino acid. Assume we are starting with the insertion. That means adding a nucleotide base. So assume now we have this sequence: adenine, thiamine. Adenine, thiamine will represent a particular amino acid when we are reading it. For example, valine. Now I'm going to do insertion. Insertion is introducing a nucleotide base between the sequence. For now, between adenine and adenine, I add guanine. So by so doing, when we are reading the sequence, it will not be adenine, thiamine, but it will be adenine, guanine, adenine. So our new sequence here will be adenine, guanine, adenine. Adenine, guanine, adenine is different from adenine, thiamine. Adenine, thiamine was coding for valine, but here adenine, guanine, adenine will code for a different amino acid. For example, valine. For example, alanine.
When you are making a protein in position number 1, which was supposed to be placed valine, as the sequence tells us that it was supposed to be adenine, thiamine, so that to place valine in position number 1, because we have introduced a nucleotide base in between the nucleotide bases, that is we have doing insertion. Now the reading will be adenine, guanine, adenine, which will code for alanine, and then instead of putting valine we put alanine. This will change, will make an abnormal protein. Of course, again, just remember, in sickle cell anemia, just replacing glutamic acid by valine in position number 6, you lead to sickle cell, meaning that the protein have a specific sequence of amino acid which should be arranged. Changing amino acid or changing the position of the amino acid, you lead to abnormal protein. That was about the insertion. Now let us have a look on deletion. In deletion, what I'm going to do, it is the opposite of insertion. In insertion, I introduced a nucleotide bases, but in deletion, I'm going to remove. So assume I have adenine, again, adenine, thiamine, guanine. So our sequence, when we are reading it, we have to read adenine, thiamine. Adenine, thiamine, of course, that assume it will denote valine. It will denote valine. Amino acid, which is valine. Now what I'm going to do, I'm going to delete, to do deletion. I'm going to remove thiamine. When I'm going to remove thiamine, when you are reading the sequence, it will not be adenine, thiamine. Instead, it will be adenine, guanine. Adenine, guanine, this will denote another amino acid, for example, alanine. So in position number 1, where I was supposed to place valine, I'm going to place alanine because I have changed a reading sequence. Therefore, whether you are inserting or you are removing, you are going to change a reading sequence, and by so doing, you will change amino acid. That's why we say genetic disease, they can have mutation which are due to frame shift. You are changing the reading sequence. You are changing the reading sequence by introducing or by removing the the nucleotide base. So when you are adding, you change a reading sequence and substitute amino acid with another, giving abnormal protein. When you are deleting, or again, you change a reading sequence, giving another amino acid, and therefore giving abnormal protein. So the classical example of the disease which fall under category of gene mutation, these are the what we call Mendelian disorders. Remember, in secondary school we used to call Mendelian disorders. Most of the conditions are familial, and they are inherited and hence familial. Most of the condition which cause gene mutation, either frame shifting or point mutation, most of them they affect sex cell. That's why they are hereditary, most of them, and hence familial. Familial meaning they can be transmitted from one generation to another. But to insist, if they affect somatic cell, they will not be transmitted. I insisted on hereditary and familial because the most hereditary disease are due to gene mutation. Okay, gene mutation, they can further be classified into 3 patterns, or they can occur in 3 patterns. We can have those which are autosomal dominant, or autosomal recessive, or sex linked. Let me explain this properly. Autosomal dominant and autosomal recessive, of course, these are the genes which occur in somatic cells. In sex linked, this occur in gamete sex series. So let us start one by one. We are going now to explain autosomal dominant and autosomal recessive. Remember the word dominant. Dominant, this is an allele. When it is present, it suppresses other allele phenotypically. Recessive, that is when it's present, it cannot express itself until there is another recessive. If we press recessive with the dominant will manifest phenotypically. It will suppress recessive. For the recessive to express phenotypically, they need to be recessive. So I hope you understand that. Now let us start to look for autosomal dominant. We say in autosomal dominant, this is a type of inheritance. In this type of inheritance, the mutated gene that determines the abnormal phenotype is a dominant that codes for a normal development. Therefore, presence the abnormal gene inevitably lead to the disease. The disease appear in each generation. It will appear in each generation. Why? Because it is a dominant. It is a dominant. Example of this are example of autosomal dominant disease. We have the Marfan syndrome, we have the retinoblastoma, we have the osteogenetic imperfecta. Retinoblastoma, remember, this is the autosomal. This is the autosomal dominant in somatic cell. Which type of somatic cell? Retina. So you have a mutation in the retina. Remember we said somatic cell gene mutation, they lead to either congenital abnormalities or cancer, malignancy, exposing you to have malignancy. So retinoblastoma is a malignancy of the retina. You have a mutation, gene mutation in autosomal dominant, leading to cancer in the retina. I will show you the picture. Okay, osteogenesis imperfecta. Osteogenesis imperfecta, this means there is abnormal development of bone. Again, this is autosomal dominant. Neurofibromatosis. Neurofibromatosis, of course, I will show the picture. This is again benign growth of fibroblast of the neuron. And the achondroplasia, this is the failure of development of cartilage. All of these are examples of autosomal dominant. Of course, you can just say this is the osteogenesis imperfecta. Of course, this is Marfan syndrome. You can see it, and this is neurofibromatosis. You can just Google the pictures in Google. You can just see the picture in Google, or you can just see them in YouTube. This is neurofibromatosis. We have type 1 and type 2. This is Marfan syndrome. This is osteogenesis imperfecta. That's how the patient looks like. Okay, coming to recessive, remember recessive cannot express itself phenotypically when there is a dominant. That means to have a disease in recessive autosomal, there must be recessive for the disease to occur. When there is a dominant, recessive will not express itself. Therefore, the patient will not show the disease, but it will remain the carrier. That's why I say heterozygous carriers. Both male and female, they may carry the genes. There is a 50% risk of the gene will be passed by child in accordance with the first Mendelian laws. Example of this, we have the sickle cell anemia. Remember, we said sickle cell anemia, this is gene mutation due to point mutation, which lead to substitution of glutamic acid in position number 6 by valine. It is example of recessive. That means for the disease to manifest, there must be recessive and recessive. If there is a dominant, there will be no recessive. Thalassemia, of course, this is the abnormality in beta chain of the hemoglobin. They lead to anemia, hemolytic anemia. We have congenital adrenal hyperplasia. You have hyperplasia of the adrenal gland. This also is this also is recessive, autosomal recessive. We have metabolic diseases such as cystic fibrosis and phenylketonuria. Phenylketonuria, this is a disorder in the enzyme which is responsible to metabolize phenylalanine, which is amino acid, leading to the presence of phenylalanine in urine. We say phenylketonuria. Okay, let us have a look of sex linked. Sex linked, we said these abnormalities which happen in sex cell or germ cells. Remember, we have X, which denote for female, and we have Y. So when there is XY, the baby will be a boy. When you have XX, the baby will be female. So remember, Y is very small, it does not carry these genes. So the one which carry genetic component is X. That means when X is mutated and the baby is Y, that means a boy, that is all boy will have the disease, because whether it is dominant or it is recessive, it will express phenotypically, because the Y cannot do anything, but in female it must be dominant. When it is dominant, the disease will manifest phenotypically, but when it is recessive, when one gene is mutated but the another is not mutated, these female babies will not have the disease, because the dominant is not mutated, and but the recessive is mutated. The recessive cannot suppress the dominant, so the baby will remain the carrier, but it will not have the disease. So to insist, when we say sex-linked genetic disease, they affect all boys. All boys are affected. Why? Because in the Y there is no genetic component, so it cannot suppress the mutated X gene. But in female, it will depend whether the mutated is a dominant or recessive. If it is a dominant, it will express phenotypically, but if it is a recessive, it will not express phenotypically. So female will remain carriers, while all boys will have the disease. Okay, example of this are Duchenne muscular atrophy. Of course, you have muscle atrophy. We have hemophilia type 1 and type 2. Of course, this is bleeding disorder. We have chronic granulomatous disease. Remember, when chronic inflammatory, when you said granulomatous. So we have glucose 6 dehydrogenase deficiency. Glucose 6 dehydrogenase. This is an enzyme which is responsible for metabolism of carbohydrate. So you have deficiency in this enzyme. Again, diabetes insipidus. Diabetes insipidus, this is diabetes. Remember diabetes from excessive urination. Excessive urination. Insipidus means tasteless, while mellitus means sweet. In diabetes insipidus, we have excessive urination, which is tasteless. This occurs due to deficiency in antidiuretic hormone or insensitivity of the receptor of the kidney. A receptor of antidiuretic hormone are insensitive. When the problem is antidiuretic hormone deficiency in the posterior hypothalamus, in the hypothalamus, this is known as craniogenic type. Or when the problem is the receptor in the kidney are not responding to antidiuretic hormone, this is nephrogenic type. Again, in diabetes insipidus, the patient presents with excessive urination, which is tasteless. Note, there are very few X-linked dominant disease, and they are much less common than a disorder arising from autosomal mutation. When we are saying there are very few dominant, meaning that most of the sex-linked genetic mutation are recessive, and this leads to all boys to have the disease and not female. Okay, after that we have finished to discussing genetic disease which are due to gene mutation. Now we have to discuss genetic disease which are due to multifactorial inheritance. We'll continue with this in different lectures. Thank you very much. So just have a review of what we have discussed. Make sure to subscribe and have a notification on, and again make sure to share this to your fellow students.